Good morning again. I'm always Andrea Mazzucato from the University of Tuscia in Italy. And to continue our course on tomato phenotyping, this is a short video about traits that can be taken in the green growth of the fruit. The fruit is the result of the growth of the ovary following successful pollination and fertilization of the flower. These events give the positive signal to the ovule to develop into a seed and to the ovary to develop into a fruit. This phase is called fruit set. The green growth of the fruit underlies a phase of active mitosis, with the size increase mainly due to cell division. This is followed by a prolonged phase of cell expansion. And when the final size of the fruit is reached, in crops like tomato, we refer to this stage as a mature green stage. The main trait that can be scored at this stage is about the so-called green shoulder. Wild-type tomatoes show a color with more or less intense green color around the peduncular side. This color is called the green shoulder, and it functions as a protection against strong sunlight and as a tissue with increased photosynthetic activity with positive qualitative effects on fruit composition. Mutants lacking the green shoulder, called uniform mutants, are preferred especially in processing tomatoes because they avoid color defects at maturity. These mutations, however, include a decrease in fruit nutritional value. The 21st trait in the CPVO protocol distinguishes varieties with or without the green shoulder. The protocol underlines the importance to use reference varieties, as the phenotype is prone to environmental effects. The CPVO protocol gets even more precise, with the trait number 22 describing the extent of the shoulder when it is present. It distinguishes 4 classes, ranging from very small to large. In the large class, the green shoulder encompasses up to the entire peduncular half of the fruit. In addition, trait number 23, always dedicated to genotypes having the green shoulder, defines the intensity of the green color of the color, being light, medium, or dark. As for the other traits related to the green shoulder, the protocol recommends to take comparisons with the reference varieties, as the trait may be influenced by environmental conditions. In our screenings of large populations, we usually give a single green shoulder evaluation, which comprises the absence of the green shoulder, the uniform phenotype, or in case of presence, the distinction into 3 levels of intensity variation, which are light, medium, and dark green. This is generally estimated by comparisons within the population, that generally presents samples showing all variations of the trait. Trait number 24 in the protocol takes into consideration the intensity of the green color of the fruit, outside the green shoulder of course. And the 5 classes are considered in the CPVO scale from very light up to very dark. In our screenings, we use a slightly simpler scale of 4 classes, the lighter named whitish and the darker dark green. Tomatoes having dark green scores for this trait usually correspond to genotypes carrying high pigment mutations. There is a further possibility that protocols do not take into consideration: genotypes harboring combinations of the anthocyanin fruit and the ateroviolaceum variants have immature fruits with dark purple or black skin due to the accumulation of anthocyanin pigments. The last trait mentioned by the CPVO protocol for the green fruit traits is trait number 25, and it is related to green stripes. A single gene mutation affecting the tomato agamus-like 1 gene is responsible of the so-called striped phenotype. This trait is typical of few varieties or genotypes like green zebra and tigerella, which are mentioned by the protocol, and is detectable even in the ripe fruit. Being due to a single gene and not showing large phenotypic variation, we did not adopt it in our screenings, but just recorded its occurrence when it was evident. So this is all again for the first fruit traits, and I wish to thank again the local organizer, the Maritza Vegetable Crops Research Institute, the Arneston Project and the Roxy Coast Action for supporting the course. And I thank you for your attention, and I wish you a lot of fun with fruit phenotyping.